stop. And I think so. There's been 119 Nobel Prizes between the 63 specialties in medicine, and 13 of them have gone to immunology. So it's had more than its fair share of Nobel Prizes, more than its fair share of incredible discoveries that have been made. But it is quite a complicated sort of body system, and the reason for that is because you kind of feel like you need to know all of it in order to understand how it fits together. It's really how it fits together that makes it work, and that's quite remarkable about it. But it means you've kind of got to take in a lot of information before it starts to click. So this will be a slightly in-depth session, and so I am expecting there to be lots of questions. So please do feel free to interrupt me while we're going through. If something I've said isn't clear, stop me before I move on to another slide, or put questions into the chat, and I will stop regularly to see what's been going on in the chat before I move on to anything. I do also just want to give a slight trigger warning that if anyone has something called trypophobia, there are a number of images in this that might feel a little bit triggering. So you might need to look away or close down the slides if those at various points. So just to quickly recap what you had on Tuesday with Valerie, where she was talking about microbiology. There are four main pathogens main groups of pathogens that tend to affect both humans and animals, actually. And in temperate, high-income countries like the UK, we're mostly concerned with bacteria and viruses. Funguses and parasites don't tend to be as much of a problem here, but they have, of course, affect many people over the world. Many of the neglected tropical diseases are parasites. Malaria, which is obviously one of the biggest killers in the world, is a parasite. And it's well known that, during the kind of colonial era of the British Empire, they called the tropics the white man's grave, because they would be exposed to so many people infectious diseases that they wouldn't encounter in the UK. So we're going to focus on bacterial and viral defenses in this session, but this will be scratching the surface of all the many things that the immune system does. Just to quickly recap cell structures, I'm sure you'll all remember from GCSE biology that the mitochondria are the powerhouse of the cell. And I just wanted to highlight a couple of extra structures that are important to be aware of. Obviously, the nucleus is where all the DNA is stored, and that's extremely important that's involved in how proteins are produced. A lot of the immune system focuses on proteins and on the information for those proteins. So it's just important to bear in mind that a lot of these cell structures from the nucleus onwards are about producing proteins. And I also wanted to mention the lysosome, which is it's essentially just a bubble inside the cell, which is designed to break down waste products, sort of proteins that have gone wrong, or, as we'll come on to in a bit, infectious diseases, infective pathogens. And finally, so what we'll go through in this session will be the three layers of the immune system. So that's firstly the barriers of the barrier protection, skin and mucosa and other barriers. We'll go through the innate immune system, which is the part of it that just works all the time on everything. And then we'll go through the adaptive immune system, which is the part that when you get an infection starts to respond after a week or two, and starts to adapt specifically to what that infection is. So in the first image, we've got the different layers of the skin. This second image is a macrophage, and we're going to come on to what macrophages are, but you can see it eating these different bacteria, these bean-shaped bacteria, pulling it into the main cell. And on the right is actually a picture of a leukemia. So it's a bone cancer which is formed from certain immune system cells, and it's also being attacked by other parts of the immune system. So firstly, barrier immunity. That's our skin, that's our mucosa, which is what you've got on the inside in your mouth and elsewhere. And this is by far the most important part of the immune system. It's what prevents the majority of infections. The skin is composed of, there's three main layers. The epidermis, that's this top part. The dermis, which is this living, more active part in the middle. And the subcutaneous, which is where there's more kind of support structures. And the important thing with the epidermis, this top area here, is that the top layer is actually dead. It can't be infected. Bacteria and pathogens can't get into it because there's nothing alive in there. It's just dead cells that are embedded in a protein layer, kind of almost like a cement in place. It's constantly shedding. There's constant movement upwards of these living basal cells that are moving upwards, shedding, and the top layer of skin is dying and coming off. And you've got lots and lots of other cells inside that. You've got arteries supplying it. You've got nerve endings, and you also have some other immune system cells which aren't barrier protection. And that's important to note that there's all of the immune system is kind of integrated into each other. But although this is primarily a barrier, we'll come on to how there's other elements of protection built into your skin as well. Mucosa is what you have. That's the kind of that's the outer layer of your body that's on your lips, on the inside of your mouth, all the way through your guts. On the inside of your eyelids. It's kind of it's got fewer layers than skin, so you've got just two layers really. The squamous epithelium, that's the top layer, and that's alive, unlike the top layer with skin, which is dead. And then you've just got connective tissue underneath, which has all those support structures. And mucosa always secretes its kind of the wet layer that you've got on top, the mucousy layer, which is often a low pH, between 3 and 5. So, for example, your tears are quite acidic, and other kind of those sorts of secretions, and that's designed to stop bacteria from growing, as well as attack other pathogens, and to prevent them from kind of getting embedded. And we can talk later about various ways that certain pathogens get around these techniques, the acid, the kind of mucus layer that traps them, in order to burrow into the body and cause an infection. Are there any questions at this point?
Nothing on the chat, Neil.
Lovely. Thanks, Joe.
Oh, apart from Paul Fisher's daughter who's joining in with some homeschooling.
Excellent. I'll have to raise my game then. So coming on to the innate immune system, this is that active part that you've got lots of cells fighting infection all the time, but they aren't specific to any particular infection. So the first part of this is going to be the process of phagocytosis, about cell eating other cells. And what this video shows is it's a macrophage which is eating bacteria, and it literally, as you can see, it moves around the body, all over the body, and simply absorbs all of those bacteria that you can see there. There are two main types of cell that do that phagocytosis. There are the macrophages, such as this one on the left, and neutrophils. Macrophages exist everywhere in your body, from the skin to the liver to your gut to everywhere. Kind of they're already sentinel, ready, watching for anything that might be causing infection. compared to neutrophils on the right, which have got that sort of funny looking nucleus. They're in the blood. They're a white blood cell that's being pumped around everywhere. And when they receive a signal that there's an infection going on somewhere, or that there's been damage, or there's something that they need to look into, if you like, the blood vessels become very leaky and allow those neutrophils to squeeze out of the blood to go and investigate whatever's going on. And that's not just for infections. Even something like a cut or a wound, which causes lots of inflammation, is going to attract white blood cells to tidy up. And this process of phagocytosis is really important for actually maintaining huge numbers of bodily functions. They tidy up damaged cells, they can remove cancers, as well as they attack pathogens. As well as those active cell parts of the innate immune system, you've got something called the complement system, which is lots and lots of proteins that are pumped around in your blood all the time, and these proteins are always being produced, and they're going off like grenades and bombs on cell surfaces. They're always attacking and attaching themselves onto cells. Our body cells have got mechanisms for throwing them off, just sweeping them away. Either we turn over the proteins and recycle them, or we can force them off the cell surface. But bacteria and other pathogens don't have those mechanisms, and what ends up happening with them is that you get all these complement proteins attaching onto the cell membrane, onto the cell surface, and creating something called a membrane attack complex, which basically means they put a hole into it. And when you've got a hole in the cell membrane, that will almost immediately destroy the cell, because the cell is really dependent on keeping a very fine balance between what's inside the cell and outside, in terms of salts and water and sugars and fats and things. If it can't maintain that, it destroys the cell almost immediately. So putting this hole in with a membrane attack complex will destroy bacteria very, very quickly. And finally, there's something called the natural killer cell, which destroys our own cells. So what it does, it's always looking around at cells that are displaying unusual activity, whether it's a virus that might have infected a cell, or if it's displaying signs of becoming cancerous. The natural killer cells are always checking cells and looking at the cell surfaces to see if there's any signs of something like that happening. And what you can see here is a picture of a natural killer cell destroying a cancer cell. And just about now, what you see there is the cell apoptosing, destroying itself. So the natural killer cell, when it detects an abnormality, it instructs the cell to kill itself. And the cell that's killing itself literally pulls itself inwards, a bit like a prune drying out or something, so that you get all these bubbles, they're called blebs, forming, and it pulls the cell apart. And that's one of the most important mechanisms that we have in the immune system, is to tell cells to kill themselves if they aren't behaving correctly, which causes them to apoptose. Are there any questions on any of the innate immune system?
Nothing on chat, Neil.
Thank you. So the adaptive immune system is built on top of the innate immune system and the barrier protection, and it's by far the most complex element of our immune system, but it's also the most effective part of it. And all vertebrates, everything with a spine in the animal kingdom, has an adaptive immune system. The most famous part of the adaptive immune system is the antibody, and there's been obviously lots in the news about antibody testing, and about how we can use antibodies, and they've got a huge number of functions, both in the body and the immune system, and we also use them a lot in scientific testing. This is the most common shape of an antibody, that classic Y shape. They've got a fixed section, which is this purple constant section, and then a variable section. So all antibodies that your body produces will have the same constant region, and the variable region has the antigen binding site. An antigen is just any protein that has a kind of information signature. It's a cell signature. So all of our cells have lots of proteins on their surfaces, and lots of those proteins will be unique to certain cell types. They can form an antigen just because they're unique, they carry lots of information, and an antibody can bind onto that unique signature. So every type of antibody will only bind to a very specific antigen, to one sort of cell signature, so that it's unique to a specific bacteria, for example, or to a viral signature or something like that. And antibodies can do lots of things. They bind onto bacteria. They can bind onto toxins, stop those toxins from actually being active, from being toxic. They can make it easy for macrophages to eat up bacteria, and specifically tell macrophages and neutrophils this is a foreign thing that needs to be eaten and destroyed. There's also lots of different types of antibodies. The two most famous are the IgG that I mentioned just before, and the IgM on the right, which is much, much larger. It's 5 of the IgG antibodies stuck together. And the IgM, it's got so many binding sites, it can agglutinate bacteria together, so that they get stuck together and end up killing each other because they're taking up all the oxygen and nutrients, and also they can't move, and they're an easy target for the immune system. Now, there's also a number of other types of antibodies which we won't go into because they're more designed for parasites and fungi, and they're secreted in other parts of the immune system and have different functions. The IgM and IgG, so IgM is secreted, it's produced in the early part of an infection. This is a graph for COVID-19 as an infection, where at day 0 you start getting infected, virus particles start reproducing themselves, and they produce lots of antigen. So this blue line shows the amount of antigen that's in the body, and around day 6, day 7, the immune system starts to produce an adaptive response. So you start getting the first antibodies being produced, and those antibodies will be IgM, because all antibodies are initially produced as IgM, and then they're cut up into smaller pieces to be IgG. But that IgM happens at the start, and then it's IgG that's produced later on. So, for example, we can use this when we're doing the antibody testing to look at what kind of antibodies someone's producing. If they're producing IgG but no IgM, then they must be in this part of the curve. They must be weeks after the infection occurred. Whereas if they're producing both IgG and IgM, we know that they're actually probably still actively infected. That's not as good as, for example, just testing for antigen. So it's not necessarily a precise way of testing for whether someone's actively infected, but it is useful we can use these sorts of information to work out what's actually going at any given moment in someone's body. Now, antibodies are produced by something called B cells, which live in the lymph nodes. Lymph nodes you've got all over your body. They drain fluid from the different tissues, so they act a lot like arteries and veins, and they're a third kind of transport system for fluid in your body. B cells live in the lymph nodes because so much fluid, so many different antigens and particles will drain through the lymph nodes. It's a very good place for them to pick up if there's anything unusual going on. And once they're activated, they will start producing antibodies. B cell activation happens when it encounters a matching antigen. So when every B cell has got its own unique antigen signature, and it basically has that from the moment it's created, that it can only detect for a specific antigen. So every B cell has its one antigen, and so for any given infection in the body, you might only have 4 or 5 B cells that will detect that specific infection. And that's why it takes so long for the response to start occurring. So once a B cell is triggered by its matching antigen, it then starts to display that on its surface. And if it's activated by another type of immune cell called a T cell, which we'll talk about in a moment, it will mature, grow up, and start producing antibodies. It will also start dividing so that it produces more plasma cells, so that it can really scale up the production. And most of those B cells will only live for 2 or 3 days, but some of them live for years and years to become B memory cells, so that if you ever encounter that infection again, you've got a ready-made response, that your body knows how to tackle this. It's still got the cells that produce the right antibodies. It's got more of them than it started with before. They're already mature, so it can very quickly ramp up that response again. And that's why you don't get the same infections twice, and that's how immunity forms. Are there any questions so far?
We do have one from Valerie. Valerie, do you want to come on mic and ask? Or do you want me to ask for you?
I had 2 actually. One was from before, from the innate immune system, and does age affect the innate immune system? And the other one is about now and the B cells. If each B cell only reacts to a specific antigen, what happens with a completely new and emerging disease? I guess that the B cells recognize COVID-19, SARS-CoV-2, even though it's a new infection.
Yeah, so the way that B cells recognize antigens is actually completely at random. So you've probably got millions of B cells that will never become activated because the antigen that they recognize doesn't exist, and it doesn't exist anywhere in nature. And I think there's about 3 or 4 Nobel Prizes have been won just for working out how the immune system does this, and creates these kind of random recognition of antigens, where you've got lots and lots of genes that code for lots of different possible antigens, but also some of it is completely random. It's literally just how does the body create its antigen and cut it up and everything. So there's a huge amount of variation. There's more variation in that particular set of genes than almost any other part of the human genome. And in fact, the theories around why organisms and cells have different sexes, why are there male and female biological sex, is probably the need to create more variation to tackle disease. And there's an excellent book about that called The Red Queen, which I very highly recommend. And all of that covers why it is that disease causes us to have a process of sexual selection, and why we need so much variation in order to be able to handle that. In terms of the innate immune system being affected by age, it is affected by age, just like all different body systems. I don't know of any reason to think that it's particularly affected by age. In terms of there is some mystery as to in COVID-19 that age is a particularly prominent factor for predicting bad outcomes, more so than we might have expected. And that's actually probably more to do with the adaptive immune system, that sometimes it can encourage too strong an immune response. And that might be what's going on partly with COVID-19, with people who become very sick, is that their immune response actually does damage to the body as well as to the virus. And we'll come on to that in a moment when we talk about the cytotoxic T cells. They're the T cells that actually handle viruses and cancers where there are changes inside the cell. Any more questions?
We've got one from Linda. Linda, do you want to come on, or do you want me to say it for you?
I was just wondering when Neil mentioned about the immune response in COVID patients. There has been talk about this cytokine rush, is it, or something like that? The
cytokine storm.
Yeah,
yes.
Storm.
Yeah. So cytokines are signaling molecules. They are released, for example, by the T cells, which are they have a coordinating function in the body. I'm just coming on to that next, actually, so I'll explain your question as we go along.
Okay, Neil, thank you.
Anything else about B cells or the innate immune system before I carry on?
Nothing else on the chat, Neil?
Lovely, thank you. So the way that antigens are recognized and passed around by the immune system, if you've got something like a macrophage that's eaten a bacteria, it eats that bacteria, and then it puts lots of it uses these lysosomes that I mentioned at the start, which are full of acids and break down all the different bacteria and all the waste that's inside a cell. What macrophages do is they just keep a little bit of the bacteria, some proteins, and puts it on a protein called an MHC, and it puts these on its surface and presents them to other cells like T cells, so that every time a macrophage eats a bacterium and consumes these antigens, it can show to other cells what it's been destroying and what it's fighting. And these macrophages can then go to the lymph tissue, for example, to find T cells that might match the right antigen. So T cells, just like B cells, have got a specific antigen that they uniquely focus on, and that activates them. So, as I've mentioned, plasma cells are mature B cells that produce lots of antibodies, and they exist in the middle of the lymph nodes. T cells are any cell that lives in this kind of middle section of the lymph node. There's lots and lots of different types, but basically they provide a kind of coordinating function for the immune system. They're the field marshals, if you like. They look at everything that's going on. They give instructions to other cells. One of the ways they do that is through cytokines that Linda mentioned. Cytokines are signaling molecules. All cells produce them, but T cells produce particular ones that instruct other cells where they need to go, how they need to fight, what particular kind of cells are needed. So, for example, if you are fighting a parasite instead of a bacteria, the T cells will secrete different signaling molecules that will instruct the immune system differently on how to combat that particular pathogen that it's facing. And T cells are the ones that are affected by HIV. So HIV specifically targets T cells and knocks out the coordinating function of the immune system. We'll talk about that a little bit more later, but more specifically, HIV targets CD4 T cell. There's two different types of T cell. One has that bigger coordinating function. It's called the T helper cell. And the other is the cytotoxic T cell. And that's got a different receptor. So there's the CD8 receptor and the CD4 receptor. And it's that receptor that HIV uses to get inside of the helper cells. And that receptor is designed to look at antigens. That's the receptor that it uses to see what's going on, what's being presented. And HIV, obviously, because it's looking for viruses, it's looking to see what's out there. When HIV attaches onto that receptor, it actually uses that to get inside the cell. So, see, as I showed in that previous diagram, an antigen-presenting cell, like a macrophage, will come along to a T cell. It will show the antigens to that T cell, and if it's a match, that T cell matures, and that means that the T cell that's needed to fight that particular infection is the one that matures, starts instructing the right B cell to be active as well, and it proliferates, so it produces lots and lots of copies of itself, so that it's ready to start ramping up that immune response to that specific infection. So the big problem there is it's an information problem, as I said. How does the body know to attack foreign antigens and not itself? How does the body tell if it's something bad, if it's virus, bacteria, cancer, as opposed to healthy cells? And ultimately, this is actually the big problem that the immune system faces. It's easy to kill cells. It's hard to kill the right cells. And autoimmune disease is the clearest example of this, that all sorts of diseases like even just mild allergies, all the way up to quite severe autoimmune diseases like multiple sclerosis, where it attacks nerve cells, those are all examples of where the immune system has gone wrong, and it's doing damage to the body without actually being able to get on top of a particular infection. The way the body uses is with these antigens. There's two main processes that the body is always looking for. Again, using this MHC protein, the presenting protein, which is called a major histocompatibility molecule. That's what cells always got on their cell surfaces to show to immune system cells. And every single cell in your body actually produces this MHC protein. So the immune system cells produce class 2 MHC, and all your other body cells produce class 1. Now the CD8 T cells, that's the cytotoxic cells, look for class 1. So they're looking at all of your normal body cells and looking for anything that's funny in the MHC protein, so that it can tell if there's virus or cancer or some other thing that means that body cell needs to be destroyed. Whereas the T helper cells, CD4 cells, are looking at the immune system cells to see what they've been eating, what they're fighting at the moment, to work out if it needs to start responding to that. And there's a whole process within the cell for how that's produced. So in a normal body cell, you've got virus or tumor antigens. Those are cut up, put onto the protein, onto the MHC, and then that's put on the cell surface. With the T helper cell, you've got that antigen, that bacteria or whatever that the immune system cell has eaten. that the same thing happens. It gets destroyed and cut up, and then it's mounted onto an MHC protein and put on the cell surface. And those are the receptors that the body is always looking at to decide if a particular cell needs to be destroyed, or if there's a infection or something else going on that needs to be responded to. Now, antigen-presenting cells are those macrophages and neutrophils that I mentioned earlier, and there's lots of different types of them, and they are everywhere in the body. So this is a skin stain, so it's a cross section of someone's skin, and the brown marks are immune system cells. So you can see how many of them there are in this particular area of the skin. There are just loads of these immune cells, always ready to pick up any bacteria, any invaders, and bring that back to the lymph nodes, to the immune system, to activate any B cells or T cells that might be able to support a response. Any questions?
Nothing on the chat, Neil.
Thank you. And again, this diagram just shows, it's a diagram of the skin. Again, you've got a hair follicle where you've got bacteria invading through the skin. You've got these Langerhans cells. They're a type of macrophage that are always based in the skin, and there are all these different types of immune system cell, always kind of distributed throughout the many different tissues in your body, ready to pick up on anything, because all of these different layers of immunity, the barrier, the innate immune system, the adaptive immune system, they all integrate together, and they use all the different possible strategies that they can to try and get on top of bacteria and invading pathogens, because it's essentially an arms race between the body and these pathogens. And there are a number of different ways that pathogens can get on top of the immune system, sometimes even using the immune system against itself. And TB is the most famous example of that, where TB is mostly, it's an infection of the lung, and you get, if you've got a lot of exposure to TB, it can get it gets breathed in, causes an infection, and the macrophages, which are there ready and waiting, eat these TB cells and TB bacteria, but then they can't destroy them because the TB is able to deactivate the lysosome, all the acids and kind of destructive chemicals that the TB wants to pour onto that the macrophage wants to pour on. The TB is able to stop any of that happening. And so you end up with all these macrophages that are full of TB that become firstly foamy macrophages and then infected macrophages. And those macrophages become diseased and stop functioning and cause a kind of pussy collection. As well as that, you've got the T cells all around trying to coordinate this response. And what they end up creating a kind of ring around this collection. And you end up forming a granuloma, which is the name for this whole kind of this whole collection. And that's where you get latent TB, where the TB can't actually get out. It's stuck inside this kind of protective ring. But at the same time, the immune system can't break in, it can't destroy the TB. And so really it's just being able to contain it inside the lungs. The problem with that is that sometimes the TB will break out and it will cause more infection throughout the rest of the lungs and also become infectious to other people. You can breathe it out in the air. So TB it's such a deadly disease to humans because it can inactivate one of the main ways that human immune systems destroy bacteria. Any questions?
We've got one from Mary Bailey. Mary, do you want to say it or?
Hi, Neil.
Hi, Mary.
I wanted to know, can lots of adverts talk about boosting your immune system? Is that just sales scam and trying to sell products, or is there actually anything in that?
I'm so glad you asked. So, there's a few different ways that you can boost your immune system. None of them can be bought in Covent Garden. So I think the irony of being named after one of the most classic adverts saying boost your immune system is that I'm going to be fighting against my namesake here. But there are two main ways that humans can boost their immune systems, other than just being in generally good health. One is something called passive immunity, and this is how babies immune systems are boosted. They receive antibodies when they're in the womb. So those antibodies cross the placenta and are supplied to the baby by the mum. And interestingly, this is where only IgG can be supplied to babies, because IgM is too big to cross that barrier. It's also provided in other ways. So in breast milk, there's antibodies. And the very first Nobel Prize for medicine in 1901 was actually awarded for someone who, for something called convalescent plasma, where you took the blood of people who had survived a particular disease, and then you separated out just it's called the plasma component, which is where all the antibodies are, and then you give that to people who might be sick with it now. So you actually supply immunoglobulins, the antibodies, to people who can benefit from them. The other way of boosting your immune system is vaccination. So essentially that process where you've got antigens being presented to the immune system, to B cells and T cells, we can induce that with by using sort of artificial antigens. So the flu vaccine, for example, it doesn't contain any flu. It doesn't contain any flu viruses. It just contains those chopped up bits of flu virus, the antigens, that we can present to the immune system for it to mount a response. And the interesting thing with the flu vaccine is choosing which bits we need to show the immune system for this year, because the flu adapts so quickly that immunity only lasts for about a year. And so we need to guess at what flu strain is going to be big in the next year and make sure that we've already provided those antigens to people who are vulnerable from it in order to prevent them from becoming severely unwell. Those are the only two ways, I believe, that you can, feel free, anyone else, to chip in if you know of other things that have been validated, but those are the only two ways that known in humans to boost the immune system.
Neil, sorry, just to interrupt. Ali's put his hand up at that point. So, Ali, do you want to come in?
Yeah, Neil, I mean, you've got the natural immunity, immune barriers, like, for example, mucosa and other things that can be obviously affected by your natural state. I mean, I remember when I did my immunity in the past, we did an experiment on people who are under fear. So we had the exorcists with two groups, and they produced less saliva, those who were under stress. So you could argue that if you are in a better state of being and better, you're more likely to produce more barriers to prevent accession to the body at the early stages. So definitely something in there to look after someone's health and well-being and not stress over things. That would help, definitely.
Absolutely, I completely agree with you on that. And there's lots and lots of evidence around chronic stress and other sorts of things having an effect on people's general health, and that may be immune mediated. So your general health is actually very, very important to the immune system. For example, we know with TB that rates were on the decline long before we had any antibiotics or any other treatments, and the reason for that was the improvements in housing and living conditions that allowed people to better fight infections. So there, you're absolutely right to say that people's general health and well-being is important, and that the immune system is such a complicated system that's what ends up and it takes so many resources for the body to maintain. But if you're already unwell, you're much more prone to being infected. That is different. So I want to draw a distinction there between your general health and a specific intervention that is known to improve the immune system. So if you were to take any substance and say this thing will improve your immune system, the only things that have been validated in trials have been vaccines and convalescent plasma or passive or sort of passive immunity. So in terms of what works outside of that, we don't have trial evidence to say that anything else does work, which of course is not the same as saying that nothing else works, but they are the only two things that we know of.
Hi Neil, it's Jo again. We've got two questions, if that's okay.
Yep, please do.
So I'll just quickly say them both together, if that's okay, ladies. So we've got Linda that's asked, how far are we away from a COVID vaccine? And then we've got another question as well about, how about vitamin D?
Who asked the vitamin D question, may I ask?
Savine,
right. Vitamin D is a very interesting one. I could talk for a while about that. In terms of the COVID vaccine, I know that there are there are 4 stages to vaccine trials. In terms of proving that it's safe, that it works, and that it doesn't cause lots of side effects. The stages go 1 to 4, and there are lots of vaccines across the world that are in stage 3. And there's apparently a very promising one being produced in Oxford and being trialed at the moment. So there is, and there are reasons for COVID-19 that we don't know for certain that it produces a long-lasting immunity, but there's good reason to think that it's not going to be like flu, where the immunity only lasts for a year at a time. COVID-19 it's a much more stable virus. And it doesn't adapt anywhere near as quickly as something like flu or HIV that we haven't been able to produce vaccines for. It's a good candidate, but we don't know for certain until we can actually follow people up for a long time and see if they develop immunity and trial a vaccine and see if that works. So there's a number of promising vaccines in development, and stage 3 is as far as they've gotten. There is one vaccine, I believe, in China that they're saying is stage 4, but they're only testing it on the military. And to be honest, I'm a bit skeptical of that, and I wonder if it's not I wonder how ethical the trial that's been undertaken is there. Now, vitamin D has been linked to huge numbers of different outcomes. Particularly if you read the latest scientific academy, I can't remember the exact name of the committee, but there was a report in 2016 to the UK government about vitamin D, and the amount of evidence that links it to all sorts of different outcomes is huge. Particularly if you do any kind of observational study, so not a trial, not an experiment, but if you just look at who's got high levels of vitamin D, you find that people with high levels of vitamin D tend to be healthier. But we don't know if that's because vitamin D actually causes people to be healthier, or if it's because people who are healthy also have high vitamin D. And one of the things we know about vitamin D is that levels go down when you're unwell. So it's actually very hard to work out where vitamin D works and where it doesn't. The only thing that there's kind of unambiguous evidence that it works is in rickets, where children's bones that are developing, they need both calcium and vitamin D. And if you don't have vitamin D, they develop rickets. If you do, it treats that. There are other things that it's been shown to be effective for. So very severe asthmatics, they are treated with vitamin D, and that appears to ease some of the symptoms. And that's really interesting because asthma is an autoimmune disease. So the immune system is attacking the lungs and causing some inflammation there, which stops them working as well. And vitamin D appears to treat that. So vitamin D is known to have a really complex and important role in the immune system, but it's linked to so many things that it's supposed to make better. That actually leaves me a bit skeptical. There are no panaceas in medicine. Nothing's ever that easy. So that's what makes me think that there must be. I think it will be very difficult to work out what it actually helps with. I know that there was a publication not long ago claiming that vitamin D might help with COVID-19, but I think there's good reasons to actually be quite skeptical of that, and that we don't have reason to believe that it does, although that's not the same thing as saying it definitely doesn't. So vitamin D, although it's a very interesting vitamin, and there's lots of research in it, I'm holding my breath on what's actually going to come out of that. Any other questions?
That's it on the chat, Neil.
Great. I'll just go back. Oh, can I go back to this slide? So COVID-19 as a virus, it just like HIV affects the CD4 receptor on T helper cells. COVID-19 uses a receptor called ACE2, which has a big role in things like blood pressure, for example. Lots of our drugs work on that receptor as well. But COVID-19 appears to use this receptor to get into cells and attack them. And these two diagrams show on the body where ACE is secreted, which cells in the body actually produce ACE receptors, and where COVID-19 can get in. And very interestingly, the lungs are conspicuously absent. The lungs don't secrete ACE; they don't have it on the cell surfaces, and so COVID-19 can't use that to get in to the lungs, despite that being where most of the damage is done. So I highlight that really just to show how much more complex, complex COVID-19 itself is. We don't really know how it does so much damage yet. I'm sure there are people who understand how it affects the lungs, but it's, the mechanisms of how it gets around, how it infects different body parts are very complicated and certainly far beyond what I understand. I think, testing. One last comment about testing, which was again about the different tests that we use. So there's two main types of tests: an antibody test and an antigen test. So just like the immune system detects a specific sort of cell signature through the antigen, we use similar processes to try and detect very specific sequences, cell signatures, viral signatures, to know if there's something present or something going on. So during the infection, this blue line shows the amount of viral matter that's in the body, and that's what we're testing for. Now you'll notice that the tail is much longer. On this diagram, it disappears at 28 days, but we don't really know for certain when it disappears. So although the test is good for showing us if someone has got COVID-19 in their bodies, it doesn't really tell us when it's gone, when the immune system is winning, when someone's actually well again. Whereas the antibody tests, particularly the IgG antibody test, that does tell us how far down the line they are. By the time you've got high levels of IgG, you're 2, 3, or 4 weeks after the infection, and probably already turned the corner. And as those antibodies inactivate the viral particles, that gives us a suggestion that people might be both immune and no longer infectious once they're testing positive for IgG. So although that's not likely, these are too imprecise to actually be used as public health strategies, it is useful to know that the reason people are infectious at the start before they've got symptoms is because that's when the virus is replicating in the body. Once people start getting symptoms, that's actually more to do with the immune response doing damage to the body in the process of getting rid of the virus. And then once the adaptive immune system has kicked into gear and started picking up all the virus using antibodies, people are no longer infectious at that point. And in general, people are unlikely to still be infectious 2 weeks after they're exposed to the virus. So, but again, those timings are imprecise, but most of the infection happens early on, before the immune system is kicked into gear. So it's early here that most of people are most infectious. Finally, just a quick comment about how we do that testing. We use something called reverse transcriptase. So that basically makes a copy of the virus RNA, which is almost exactly like DNA, except it's not. It's got a very slightly different chemical signature, and that's we create copies of the RNA millions and millions of times until we can find the exact cell signature, the exact virus signature that we're looking for. And if we're able to find that virus signature, then the test comes back positive. But it's using that RNA. And finally, there is one other type of test that we can use. This will be the kind of thing that we use for the antibody test, although there's a number of different types. And it's called ELISA testing, which is about using antibodies to detect if there's a particular molecule. Because antibodies have that really specific binding, they'll only bind onto really specific sort of protein signatures. We can use those antibodies to see if there's something present in the body, or in lots of other situations. And if the antibodies do bind onto something, they'll light up under green light, and so we're able to tell if there's that particular kind of viral signature, for example, or if there are particular antibodies present, or it's a very general use test that is used all throughout biochemistry. Are there any questions?
Nothing on the chat, and if anybody wants to turn their mics on to ask questions, they can. Oh, Richard Cooksey, do you want to say your question, or are you happy for me to say it?
I was just thinking, is there any way the blood supply is okay? Can they test for COVID? I suppose just thinking what you were saying, really.
Sorry, Richard, would you mind just repeating your question?
Yeah, sorry, my microphone's not close. Yeah, is there any blood, or any, if people donate blood to use in the hospital, and they make blood products and use it in operations, can they accurately test for COVID in those blood supplies with the tests we have and that sort of thing?
Yes, so they can do this one called RT-PCR, reverse transcriptase PCR, which detects any viral RNA in the blood. Now, and actually, we do use blood donations a lot. We test them for COVID, not just to prevent it from being passed on in donations, but also to see how many people have got or have had COVID. So when you've got all these blood donations, which are basically blood samples that are ready and waiting that people have happily given, and these people are normally fit and well, they don't have any symptoms, so they're a good test for us to see if they what the general population level of COVID-19 is. Obviously, the problem with that is that the people who go and give blood aren't necessarily just like the rest of the population. There might be differences about them compared to other people. So we don't know for certain if it's a good sample, if it's reflective of the population. But it is we do use testing of blood donations both to protect from passing it on, but more importantly, actually, as surveillance, because it's very unlikely you would pass anything on in a blood donation anyway.
Oh no, thanks for that. No, I hadn't thought about that way of finding out about how many people have it, but as you say, it wouldn't be very robust about the sample and that, maybe, because there might be certain types of people might give more and that sort of thing, maybe.
Yeah, of course. Any more questions?
Nothing on the chat, Neil. Just everybody saying that they're finding the session absolutely fantastic.
Oh, lovely. Thank you very much. That is the end. I'll stay around if there are any more questions.